Welcome to Ajat Bioacademy. Today I will talk about topoisomerase enzyme. Basically, bacterial topoisomerase. That means prokaryotic cell topoisomerase. You know, it has some difference with the eukaryotic cell, as some antibiotic will work on topoisomerase enzyme as a target molecule. So, my main focusing point is bacterial topoisomerase enzyme, and I will try to cover what is topoisomerase enzyme and what are the types, what are their roles, and how it become a target molecule for some antibiotics. Okay, so first of all, the terminology is topoisomerase enzyme. Okay, so topoisomerase, from the name, if you just look at closely, then you will get two things. One is topo, another one is isomerase. Isomerase is very silly things, that means isomerism, and now topo comes from topology. You know the topology. So what does it mean, topology? Actually, topology means the study of continuous changes of something, the shape and size, the geometrical changes. That is called topology. So some enzymes will work on the changes of the DNA, genetic material. That is, those enzymes are called topoisomerase enzyme. That means it will work on the changes of the topology of the DNA. So during replication and transcription, DNA must undergo the changes to make its copy or to transcribe into RNA. So that changes can be replication fork, and before and after some unwanted changes, that means abnormal changes, and that will be the hindrance of the replication or transcription, and some enzyme will work on it to just reverse that condition to facilitate to replication or transcription. That means topoisomerase enzyme is very important for replication and transcription of the bacteria. So now come to the point, topoisomerase enzyme. So firstly, topoisomerase enzyme, already I said, it will work on the structural changes of the genetic material. So firstly, before going to the topoisomerase, you must know about the basic structure of the genetic material, that is DNA. So you know as well, but related to that, how it will become supercoiling and how it will become the very suitable for replication and transcription. That is the point. So firstly, you know the DNA is right-handed structure, right-handed double strand helical structure. So that is, you know. Okay, so if you think the orientation of this DNA is like that, that means the orientation is clockwise. If you just consider it as like this is your right hand and this is your thumb and this is like that. So when you will think in that orientation, so your helical structure would be like that. That means right-handed. The orientation, just according to the clock, that will be the clockwise orientation. You can think the right-handed helical structure. So DNA in generally, normally right-handed double helical structure, double strand helical structure. So you know the helix means the S-like structure, and double strand helix, that means the S structure and the helical and then helical. So double strand, and why it is called right-handed? The helical orientation is right-handed. That is like that. Okay, so this is the way. But when, if you try to unwind it, then what will happen? When you will just snatch it in that direction and that direction, then what will happen? It will unwind, and in the backward area, the tangle, twist, or torsion will form. That means it will compact. Suppose if you just unwind a rope, then you will find the answer. In the backward area, it will twist it within itself. That means normally this is like a coil, and it will again changes like a super coil of a coil. If the direction just like the normal DNA, that is right-handed, then you can say this is the positive supercoiling. Shortly, I will explain it. That means if you just try to unwind it, then what will happen? That will form a fork, and in the backward area, that will form like that, and finally that will form like that. That means okay. So here the orientation is same in right orientation. So you can say that is the coiling compactness, and a tangle will form in that same direction. So you can say that is the positive super coiling. And that is the problem, because when you will just pull the two strand, or you can say when helicase enzyme will work on it and it will unwind, and then in backward in upward area, that means upstream area, tangle, or you can say the compactness will increase more and more, the torsion will form, and finally tangle or knot will form. Then what will happen? It will coil of within itself. Then what will happen? It will form a supercoil, and that supercoil is positive supercoil because the orientation is same like right-handed helix. Okay, so in the right-handed direction, if coiling happening, then you can say that is the positive supercoiling. Okay, and this is the problem. So some enzyme will work on it to cut that strand, and it will just again reverse the normal condition, and then finally unwinding by helicase, and it will go on the replication and transcription. Okay, so this is the positive supercoiling, and you can say negative supercoiling also be possible, because when the topoisomerase will enzyme will work on it, and it will just make it negative supercoiling. So what does it mean, negative supercoiling? And now I will just roughly try to give you some idea. That is, your orientation would be like that. Okay, so your orientation is like that. That means anti-clockwise. This is the clockwise, that is in that direction, and that is the anti-clockwise, and the direction is left-handed, and you can consider it as like, okay, in that way. That means that is like, so this is the left-handed helical DNA, and this is the left-handed helix. Okay, so this is the left-handed helix, this is right-handed, and this is left-handed. Okay, so when you will unwind it, and if the twisting occur, or compactness, or the coiling will happening more and more in the opposite direction of normal DNA, then you can say this, that would be the negative supercoiling. Negative supercoiling. Both are problematic. Excessive negative supercoiling also problematic, and positive supercoiling also problematic. So some enzyme will work on it to make the positive supercoiling into negative supercoiling, and then helicase enzyme will work to unwind it. And if excessive over negative supercoiling happening, then that will also hindrance, or that will also inhibit the replication or transcription process. Okay, so basically negative supercoiling is applicable for transcription, that's backward area, and also over activation of topoisomerase enzyme that can cause negative supercoiling. So both are problematic. Okay, so now I will explain where it will form the supercoiling and how it will become resolved by the topoisomerase, and what are the types of topoisomerase enzyme. So this is the basic structure of DNA, right-handed and left-handed, and this is the positive supercoiling, negative supercoiling. Actually, negative supercoiling is desired, one, to facilitate the replication and transcription, but when it will be over expression or more supercoiling, negative supercoiling, that is also problematic. Okay, so now come to the types of supercoil topoisomerase. So now the topoisomerase types are basically 4 types. One is topo 1, second one is topo 2, third one is topo 3, and last one is topo 4. Actually, we are talking about the bacterial topoisomerase. Okay, so one after another, I will try to cover its location, its function, role, and possible drug target. Okay, so first of all, topoisomerase 1. So topoisomerase 1, the characteristics of topoisomerase 1 is it is basically monomeric. Monomer structure. That means long chain of amino acid that has a C terminal, N terminal, and core domain structure, and that will form an enzyme that is called topoisomerase 1, and that will cut or cleavage a single strand. That means it will cut or cleavage single strand of DNA. Okay, and it has no need any energy, no ATP needed. Okay, and mainly it will work on during the transcription process. Transcription process. That is the star mark. That is main working area during transcription, and also replication stage. Okay, so how and where I will just show you later on. So this is the basic criteria of topoisomerase 1 enzyme. That is monomeric, and it will cut a single strand. It has no function with the ATP, and transcription. During transcription, it will show its activity, and during replication also. So now how it will work. So come to the point here. So that is, suppose if you think, if you just unwind during transcription, so I will focus you, that is, you can say, consider as this is the DNA, normal DNA, and this is your helical structure, DNA structure. And if some particular gene, suppose that part of gene will be transcribed into mRNA, so that part will be transcribed. So what will happen? A transcription bubble will form. Some polymerase enzyme will bind, and it will form a transcription bubble. Okay, so now how it will form a transcriptional bubble? Transcriptional bubble. This is the transcriptional bubble. Then what will happen? The one strand, there's the template strand, or you can say antisense strand, will make a particular RNA. Suppose from that strand, it will produce a mRNA. Okay, that is called template DNA strand. So this is the double strand. This is the transcriptional bubble. So now, where is the role of topoisomerase 1? Basically, in the backward area, when the direction of the transcription will happening 5 to 3' alloys. So when transcription happening by polymerase enzyme, then in the backward area, the DNA will be negatively supercoiled in the backward area. So then what will be the problem? Because messenger RNA will just forming, and it will just go forward. That is the problem, is the polymerase enzyme will not bind properly with that area. That is the problem. Another problem, it will change the orientation, and finally it will hamper the transcription. Supercoiling, negative supercoiling. So already I said negative supercoiling means the left-handed helical twisting or more compactness of the DNA, and that will form a coil of a coil. That means supercoil. That is negative supercoil. When positive supercoil will be formed in the front of replication fork, that is the problem, and some enzyme will work on it to make it the negative supercoil, that is the desired one. But when negative supercoiling happening more and more, then it will be the problematic and it will hamper the transcriptional process. So basically, in the backward area, transcriptional stage, when negative supercoil will form, and that will hamper the transcription. In that area, the topoisomerase 1 enzyme will work on that area. And how it will solve that problem? That is, suppose here is the uh, bubble, and here is the negative supercoil. Okay, so now it will just, suppose here it will cut a single strand here. It will cut that strand and it will just unwind from the inside to outside, and then again it will ligate each other. That means it has function for cutting and ligation also. Okay, so now what will happen? It will cut a single strand and it will just uncover it, and it will just remove from the inside to outside, and then it will again ligate. Again it will join. Okay, and it will solve the problem of the tangle, or you can say supercoil. Negative supercoil will be resolved by that topoisomerase 1. So you can consider this is a topoisomerase enzyme, and that will cut a single strand. Suppose just like that. If you cut that strand, then it will just come out from that part, and again it will be like that. Okay, so that problem will be solved by the topoisomerase 1 during the transcription. And if you consider during replication, that would be another explanation. Very shortly I will explain, although it has very similarity during the transcription, but during replication it has some distinction from the transcription. So that is normal DNA, and bacterial DNA, round DNA. Most of the bacteria carry round DNA, and some bacteria can carry the linear DNA, but mostly round DNA, and it has a single replication origin. So when during replication origin, it will work to unwind. Then what will happen? It will form a replication fork. It will form a replication fork. Okay, so that will form a replication. That will form a replication fork, and this is the replication bubble, and that area is replication fork. In two orientation, replication will happen. And when the helicase enzyme will go forward, that means in upstream, then here right-handed twisting will happening, because you are just try to stretch to strand, and in forward part it is just twisting. The compactness will happening, torsion will increase, and finally tangle or knot will form before the replication fork, and that orientation is just like the right-handed DNA. So here that will form a like structure. Okay, so very compact and finally tangle. So this is called positive supercoiling. Okay, and that is the very big problem for the replication. So you must just solve the problem. So topoisomerase 1 enzyme has only capacity to cut a single strand. So it has no capacity to solve the problem of supercoiling. That means major knot, major tangle, it has not possible solved by the topoisomerase 1. So what will happen? In similar way, it will cut a single strand. Suppose here it will cut the single strand, and it will make it inside out, and it will just resolve the uh, torsion, then facilitated to work of helicase enzyme very easily. Okay, so the function is topoisomerase 1 will work on the positive supercoiling, and it will turn into, firstly it will turn into negative supercoiling, and then helicase enzyme will easily unwind it and replication will go on. Okay, so topoisomerase 1 is basically work during transcription and also can be responsible for uh, making it negative supercoiling during transcription. Okay, so this is topoisomerase 1 basically. Now, so topoisomerase 2 is topoisomerase 2 is another name is gyrase enzyme, and sometimes it is called gyrase enzyme. Okay, so that topoisomerase enzyme, it has some characteristics. So it is basically tetrameric, and it has two subunit, dimer and dimer. So the subunit is gear A, and you can say gear B, and both are dimer. Okay, so finally that is tetrameric. Okay, and it has a capacity to cut double strand of DNA. That means it can cut double strand of the DNA, and for its function ATP is needed. Okay, ATP is important, and where it will work and during when it will work, and its main function during replication. Okay, and also so it has replication, and during replication it has main function, and also it has function during decatenation. Decatenation, that means segregation of chromosome. Segregation of chromosome. Okay, so this is the basic criteria of topoisomerase 2. So it has two subunits, and now I will show you the rough structure of the topoisomerase 2, that is gyrase enzyme, and how it will work. I will explain here now. So this is the function. So it is tetrameric, and it has two subunits, gyrase A and gyrase B, that is gear A and gear B, and it will cut the double strand of the DNA at a time, and for that reason, for that purpose, ATP is very much important for that, and it will work during the replication as well as during the separation of chromosome, that means from parent chromosome to daughter chromosome. Okay, so now I will explain with the structure. So I will just roughly, I will try to explain it. So this is one subunit, that is upper subunit, upper part subunit, and this is called gear B. Okay, and here another subunit is there, that is gear A, and within this another terminal, that means domain, C terminal domain is attached with that gear A, that is C terminal domain, and here is C terminal domain. It's very important. I will explain all the functions of it. So this is the gate A, this is the gate B, and this is the uh, C-terminal domain, and this gate name is called N gate, and this gate is name is in between the gate A and gate A and B. That is the gate name is DNA gate, DNA gate, and this gate name is called C gate. Okay, so now what will happen? This gear B part, that is upper subunit, the dimer and dimer, that means monomer and monomer, that is dimer and that dimer. So total is tetramer. And what will happen? This gear B part, it has a ATPase activity. It has ATPase activity. That means ATP will bind that arm and that arm. Two ATP are bound with the two arm, and ATPase will work on it. That means it will hydrolyze the ATP, and then it will, that is the, you can say this is the ATP, and ATP it will hydrolyze and it will form energy. Okay, and that energy will change the conformation of the enzyme, and that will facilitate to open the channel, and it will facilitate to work the all other subunits and the terminal. So what will happen? Through this gate, DNA will enter. DNA will enter. Okay, and what will happen when ATPase activity will be functioning? Then ATP will hydrolyze and it will produce energy. That energy will change the conformation of that gear B, and that changes will facilitate to enter the DNA, and the DNA will captured by the CTD, that means C-terminal domain, and that C-terminal domain will wrap up the DNA and it will facilitate to enter into the D gate, and finally your gear A will be functioning. And that gear A, it has two functions basically. One is cleavage function, or you can say cutting function, and function 2 is ligation, or you can say joining function. Okay, so already I said it will cut the double strand at a time, and already I said before replication fork the positive supercoiling will be happening, and that positive supercoiling is the orientation is like right-handed, but it is more compact and more tangle will form. Then that enzyme will be functioning on that part. That means, just I will show you very roughly. Okay, so this is the replication fork, and this is the supercoiling, and very simple supercoiling will be solved by the topoisomerase 1, but when it will be form more tangle and more twisting, then what will happen? It will be solved by the topoisomerase 2. What will happen? It will just, suppose this is here, it will bind with the topoisomerase 2 and it will cut that part. That means both strands will be cut. Suppose like that. I will show you by through my hand. Like this is topoisomerase 2 enzyme, and this is your N gate, and that is, you can say this is your CDT, and when ATP will bind and then conformation will change, the DNA will enter, and then it will go down to the gear A part, and then what will happen? It will just cut the double strand. It will form a negative supercoil, and then finally helicase enzyme will easily make it a unwinding. Okay, so what will happen? The double strand double strand DNA will cut off by the guidase enzyme, that is topoisomerase 2. Fluoroquinolone derivatives, ciprofloxacin, moxifloxacin, levofloxacin, that will work on the GIR A part. In which part? In the ligation part. It has two cutting site. One is cutting site, another one is ligation site. So it will the antibiotic will work on the ligation site. That means it will facilitate to cut the double strand DNA, and the another part of the GIR subunit will rejoin it, and it will solve the tension. Okay, but when antibiotic will inhibit the ligation, then in one part it will cut off the DNA more and more, and the fragment of DNA will be accumulated. Another part will be inhibited by the antibiotic. So the ligation part will be inhibited. In one direction cutting is happening, another the ligation is hampering. So what will happen? The DNA will be degraded and bacteria will die. Fluoroquinolone derivatives will work as like bactericidal antibiotics. Okay, so I will just give the explanation in later one, the antibiotic part. So it is basically tetrameric. It has two subunits, GIR A and GIR B. GIR B basically responsible for the hydrolysis of ATP, that is energy production, and GIR A is responsible for cutting and ligation. Okay, energy is very much important, and during that part it will basically work on the positive supercoiling to resolve it by making it negative supercoiling and finally unwinding. And another function is it will work also on the decatenation or segregation of the chromosome. That means, this is your round DNA of the bacteria, and that would be copied. So what will happen? It will copy, but it will be like form like a interlinked. That means as like a ring, one ring will enter into another. So if you want to separate it, then what to be done? You must cut a double strand of DNA, and then your DNA will be removed from it, and then ligation will ligate it, and finally two chromosomes will be separated. But if that enzyme is not working, so it will form a interlink as like a chain ring within the ring. So it will be a problem. It will hamper the replication. So topoisomerase 2 also work to cut off that area, that is double strand, and it will facilitate to separate the two round DNA. That means it will be replicated. That means it will separate the chromosome, and it will form another bacteria. It will form another bacteria. Okay, so this is about the function of topoisomerase 2, that is gyrase enzyme, or sometimes you can also say gyrase enzyme. Now topoisomerase 3. Topoisomerase 3. Actually, its function is identical of topoisomerase 1, and its nature is also like that. So it is also monomeric. It will cut single strand, no ATP needed, and it will actually work during DNA repairing, or you can say recombination. Okay, so actually during the positive supercoiling or negative supercoiling replication transcription, in that area topoisomerase 3 has no such type of role, but it has some role during the DNA repairing. That means DNA will be faulty, then it will repair. So in that case topoisomerase 3 will work. So as it has no connection with the antibiotics, so I will not give it the details. So now I will go for the another important one, that is topoisomerase 4. Topoisomerase 4. Structurally and functionally, it is very identical with the topoisomerase 2, that is gyrase. So now, if I try to explain its features, its function or characteristics, then it is also tetrameric. Tetrameric. It has two subunits. One is per C and per E. Both are dimeric. So finally tetrameric. ATP needed. It will cut double strand of DNA. Okay, and it will work basically during segregation of chromosome, or you can say decatenation. Okay, so just like topoisomerase, but the place of work, or when it will work, during the segregation of the chromosome. Okay, so this is the difference, but different other things are mostly common. So structure is also similar like that. That means it has roughly, it has two-arm upper part, that is, you can say this is the power E, and here another part, you can think like that is power C, and this is N gate, this is your C gate, and this is your DNA gate, just like just same as topoisomerase 2. But main function difference is segregation. So when it will just, already I explained just few minutes earlier, that means this is your round DNA, and round DNA will unwind in a part of replication, and that will make a copy, and that copy must be separated from the parent chromosome. So during separation, it will just form a interlinked as like a chain. So here it will form a tangle, or you can say not tangle, that will form a interlink. That means like that. Okay, so you must separate it. So what should you do? The enzyme must be applied, that is your topoisomerase 4. So topoisomerase 4 will be functioning over there, and it will cut the double strand, and it will just make the one strand from inside to outside, and it will facilitate to separate the daughter chromosome from the parent chromosome. Then what will happen? It will just separate. Okay, it will separate. So this is the segregation of the DNA. So if that enzyme, that is your topoisomerase topo 4, if that topoisomerase is not functioning over there, then what will happen? No segregation will form, no decatenation will form. That means it will be interlinked with each other, and no separation, no multiplication of genetic material. That means no proliferation. So that is the function of topoisomerase 4. Okay, here antibiotics like your fluoroquinolone derivatives, ciprofloxacin, oxyfloxacin, or levofloxacin, that also inhibit the topoisomerase 4, and that topoisomerase 4 will be inhibited, then finally no segregation of the chromosome will be happening, and then finally your bacteria will not be proliferated. Okay, so bacterial growth will be hampered. Now I will sum up the overall topoisomerase 1, 2, 3, 4, and then I will go for the antibiotic regarding this topoisomerase. So that is topoisomerase 1 is monomeric. It will be able to cut the single strand of the DNA, and it will work during transcription and replication, uh, negative supercoiling and positive supercoiling. Topoisomerase 2 is tetrameric, and it has two subunit, upper subunit and lower subunit, that is grid B and grid A, and it will cut the double strand, and it will work basically during the replication, and also can be functioning for during the separation of chromosome. And topoisomerase 3, it has basically function for DNA repairing, and it is structured as like as topoisomerase 1. And topoisomerase 4 structurally similar to the topoisomerase 2, and basically it will work during the segregation, or you can say decatenation of the chromosome. Okay, so this is all about the topoisomerase enzyme. Now I will go for the antibiotics. So already discovered antibiotics is fluoroquinolone derivatives. That is the fluoroquinolone antibiotics. Okay, so that fluoroquinolone set, it has 4th generation. The first generation you can say that is Nelly Digic acid. Second generation you can say ciprofloxacin. Third generation you can say levofloxacin, and fourth generation you can say moxifloxacin. Okay, so these are the fluoroquinolone antibiotics, and it has different role, gram positive towards the gram negative. I will explain in separate video, but I will try to connect with the topoisomerase enzyme. So how it will work? Actually, those antibiotics will inhibit two enzymes, topoisomerase enzymes. One is that is topoisomerase 2, that you can say that is guidage enzyme, and another one which will inhibit the topoisomerase 4. Okay, so that topoisomerase 2 is very functioning, very prominent in gram negative bacteria, because gram negative bacteria metabolic rate is very high, because it is very much specific for the nutrient supply. It has outer membrane, and outer membrane has porine protein, and through this protein the desired nutrient easily pass through the inside, and it will use for their energy production and then for their metabolism function. Okay, so its metabolic function is very high, and it has, gram negative bacteria, the peptidoglycan layer is very thin, so it can easily separate from each other. Okay, so overall the metabolic and replication rate is higher than gram positive bacteria. So that topoisomerase 2 is very prominent in gram negative bacteria, because it has metabolic rate is high, and it has very specific nutrient supply channel, and it has very thin peptidoglycan layer. Okay, and topoisomerase 4 is very prominent in gram positive bacteria, because during segregation, because the metabolic process, that means replication process, when it will be higher, then positive supercoil will form in front of the replication fork, that means upstream area. So it's very much important to be functioning of topoisomerase 2. If topoisomerase 2 will be functioning more, then what will happen? It will be more functioning to make the positive supercoil to negative supercoil. Okay, but when, if you apply the antibiotics and if it will work on the topoisomerase 2, then your positive supercoil will never be negative supercoil, will never be resolve the problem. That means tangle. That means it will never resolve the knot, never resolve the tension or compactness of the DNA. Okay, so already I said what will happen. Your topoisomerase 2, the guidase enzyme, has two functions. One is cleavage function or the ligation function. But on which part of the guidase enzyme of topoisomerase 2, that will be inhibited. That is, ligation part will be inhibited by those antibiotics. Okay, so gram-positive bacteria, it has very thick peptidoglycan layer, and during separation, it will hamper the segregation of the chromosome. Okay, so topoisomerase 4 is it's considered very strongly during the gram-positive bacteria. Okay, so what would be the mechanism? That mechanism is it will inhibit both topoisomerase 2 and both topoisomerase 4. It will be inhibited by those antibiotics. And topoisomerase 2, already I said, it has a GIR F part, that is lower part of the lower subunit of the enzyme, and it has a cleavage function, and another one is ligation function or joining function. So it will, that those enzymes will inhibit actually that part, ligation part. The cleavage will be continued and the ligation will be inhibited, and finally bacterial DNA will be cut off and bacterial DNA will be accumulated, bacteria will destroy. Okay, and when topoisomerase 4 will be inhibited, then it will never be segregated. The chromosome will never be segregated, so bacteria will not replicate. So those antibiotics will inhibit the bacterial growth, as well as it will just destroy the bacteria. Finally, it will work as a bactericidal because it will mostly work on the topoisomerase 2 and GIR subunit. It will bind with that and it will inhibit the ligation, and it will continue the function of topoisomerase 2 cutting cleavage function will be continued, and finally DNA will be segregated, DNA will be degraded, DNA will be fragmented, and finally bacteria will die. Okay, so this is all about the fluoroquinolone derivatives antibiotics and their classification and their function, their mechanism of action. Okay, so I will make a separate video on the fluoroquinolones, their specific difference of the individual generation and basic drawbacks. Okay, so that would be my another video. So thank you very much for your patience hearing. If you think my video is share worthy, then you can share and you can subscribe my channel. Thank you very much.